Hi, my name is Heather Schofield, and today I'm going to talk about work from Phil Bednar's lab at the University of Michigan, showing that BMI is required for human pancreatic cancer growth, and it does this by controlling cell metabolism. Previously, we showed that BMI is required for the initiation of pancreatic cancer in mice. So using mouse models, we saw that those animals with a KRAS mutation but without BMI expression were not able to initiate pancreatic cancer, and so told us that BMI is required for this first step of pancreatic cancer. From here, our next question was, is BMI required in later stages of pancreatic cancer growth? In order to ask this question, we used CRISPR to knock out BMI in human pancreatic cancer cell lines. And then we used these cell lines and grew them either subcutaneously or orthotopically in immunocompromised mice. And when we grew them subcutaneously, we saw that the wild-type pancreatic cancer tumors grew quickly. However, the tumors lacking BMI expression were much smaller. This was reflected in the resulting histology, where in the wild-type tumors we could see numerous tumor cells, and in those lacking BMI expression, we saw mostly fibrous material with small nests of tumor cells. We saw similar results in the orthotopic experiments, where the BMI knockout tumors were quite small at the end of the experiment. And so overall, this data told us that BMI is required for pancreatic cancer growth, so later stages of pancreatic cancer. What we next wanted to ask is, what is BMI controlling in order to be required for pancreatic cancer growth? And so to ask this, we did RNA sequencing of our pancreatic cancer wild type or BMI knockout cells and analyzed which pathways were changed. When we looked at the pathways, one of the things that stuck out to us were many changes in metabolic pathways, and specifically in those that control glycolysis. And we saw changes in expression of every gene in the glycolysis pathway when BMI was knocked out. What we wanted to do next is see if BMI plays a functional role in metabolism in pancreatic cancer. And so to do this, we performed metabolomics and then analyzed the top 10 altered metabolic pathways. And once again, we saw that glycolysis was one of the top altered pathways when BMI was knocked out. And this is reflected here in this heat map. And so this told us that BMI has a functional role in controlling glycolysis and the progression of pancreatic cancer. So in this work, what we've shown is that BMI is required for pancreatic cancer growth, so this later stage of pancreatic cancer, and that it does this by controlling metabolism and glycolysis. And with that, I'd like to thank my funding.